The human immunodeficiency virus, or HIV, causes AIDS. There are currently treatments for HIV, but there is no cure. Once HIV enters the cell, it hijacks cellular components as it converts its genomic RNA to DNA in a process called reverse transcription. It then integrates the newly transcribed DNA into the host genome. HIV infection occurs deep within the complex environment of the cell. To learn how HIV machinery interacts with molecules in the host cell, it is important to study HIV in a less complex environment. That is why the Sundquist Lab at the University of Utah came up with a new method to study these first steps of HIV replication in a cell-free system. Here's how it works. First, we isolate HIV virions. Next, we remove the outer membrane of the virus with a molecule from bee venom. This process liberates the cone-shaped viral capsid, which protects the viral genomic RNA inside. We tested different mutations of the HIV capsid to investigate the role it plays in reverse transcription, comparing the mutants to wild-type HIV capsids. Capsid-stabilizing mutants held the capsid together longer than wild-type, and destabilizing mutants produced capsids that disassembled more quickly. After mixing in all the components needed for reverse transcription, we incubate it for 10 hours. During this period, HIV is busy reverse transcribing its RNA into DNA. We can also model HIV integration in our cell-free system by adding cell extract and a DNA target for HIV to integrate into. In the end, we measure the DNA that HIV produced. Remember the different mutants of the capsid that we tested? Surprisingly, the capsid that supported the most efficient reverse transcription was the wild type, showing a direct relationship between capsid stability and reverse transcription efficiency. What's next for our cell-free system? Finding unknown host molecules and how they interact with HIV to enhance or disrupt infection. This can pave the way to better treatments for patients and someday a cure. If you would like to learn more, here's the reference to our paper.